Hi, my name's Kirstie. I'm a researcher in the Department of Psychiatry at the University of Cambridge and also a research fellow at the Alan Turing Institute for Data Science. I'm super excited that you're taking part in the online brain intensive, and I'm really honored to be part of this very first week, giving you an overview of reproducibility in brain science. There are so many great lectures that are going to come up over the course that what I've focused on today is really the big picture, the why, and then also trying to just give you this extra sort of boost of confidence that even if some of the exercises that you've either heard about already or that you might encounter over the next few weeks feel kind of intense and like they might be difficult to fit into your own personal workflow, I hope they're not. But in case they are, there are always things that you can do. There are things that you can start doing right now to make your work more reproducible. I always wanted to be Britney Spears growing up. And part of that is getting to wear this super cool headset. Uh, Another part of it is so that you have a really good experience of being able to hear me much more clearly. So I hope you like it. Let's get started. Okay, so reproducibility in brain science. I already told you a little bit about who I am. I work at the Alan Turing Institute, and I'm also affiliated with the Department of Psychiatry at Cambridge. And in June of 2017, I completed a 10-month stint as a Mozilla Fellow for Science. Mozilla Science Lab are focused on bringing the philosophy of open source to science, and one aspect of that is making sure that we have reproducible published research. So the definitions of reproducible and the definitions of replicable. Okay, so I've got a little grid here. For a given research question, if I give you the data that I used, the same data, and I give you the same code or the same analysis pipeline that I used, and you are able to get the exact same results as me, I would say that my work is reproducible. Now, if you've ever gone out and spoken with your friends who are non-research scientists, who are sort of outside of an academic publishing world, the idea that we don't currently require this blows their minds, as well it should. This cell, to me, is the bare freaking minimum for publishing scientific research in the 21st century. We can share the data. We can even share the data privately if there are any problems with ethics, but we can make it so that other researchers can verify our work by sharing the code, the pipeline, the steps that we took, and the data. Now, a different cell is if I give you my code or my analysis pipeline, the various steps, and you have some data. So it's an independent data set. Maybe it's some structural MRIs that you've collected as part of your study. And you want to see if for this given research question, if you take the same code as my original piece of work and run it on a different data set, do you get the same result? And if you do, then the work is replicable. And this is a really nice sort of step towards being more, I would say, reliable and definitely more useful, because it means that that particular finding that I published was not specific to my code and my data set. Now, the next cell is taking the original data, but looking to see how robust it is to different types of analysis pipelines. Now, if you think that the analysis pipeline is different, then that's not trying to address the same research question. But if you think that doing an analysis in FSL versus an analysis in SPM versus an analysis in AFNI versus an analysis in one of the NiPi family tools, if you think that that shouldn't matter to your result and you run those various different packages on the same data set and you get qualitatively the same result, if you would conclude the same take-home message from all of those analyses, you have a robust finding. And that's our step towards what I think we should really be seeking, which are generalizable research findings. And that means that instead of doing it as one set of authors on a one-off publication, we actually say we had a reproducible finding from the beginning, it replicated in a different data set, it was robust to various different labs with their various different types of customized packages, and you take home the same message. And I call that generalizable. It's not generalized, absolutely not. There's lots of other sort of aspects with who comes in to be part of our studies and things like that. But it is at least a step towards making it useful, for example, for policymakers who want to know how their decisions are going to affect, for example, the brain development of children in schools, or clinicians who are working with, in my particular area of interest, people who have episodes of depression or psychosis, and whether they will respond to treatments. I'm sure most of you have experienced barriers to reproducible research. I certainly have, and when I go around and I talk with people about my desire for reproducible research, I hear a lot of sort of pushback against it. So on this slide, I've added in a few of the common kind of arguments that I hear, and I'm going to talk you through them now. The first grouping is this problem that reproducible research, the effort that's required to ensure that our research is reproducible, is not work that is currently considered for promotion. And it's also the case that reproducible research may be held to higher standards than other research. So when peer reviewers see a publication that has no data and no code associated with it, and they let that slide, whereas they may, if there's code and data available with a publication during peer review, they may actually query that more stringently than if there was nothing there in the first place. These, to me, are hugely important barriers. They are entirely justified reasons to not want to do reproducible research. And I think that they are something that the most senior members of our communities are able to change. They are also things that the funders are able to change. So, and the journals, that it just, for me, it just has to be the case that we need to remove the possibility for promotion or for publication if you do not do reproducible research. The demographic of you, the people who are taking part in the online brain intensive, I don't think this is your responsibility. This is something that myself, as a sort of early career researcher who has a little bit of independence and my own funding, and more senior people, this is where our responsibility lies. There's another problem, which is that at the moment, journals accept work that is biased towards novel findings. And that means that, fine, reproducible research may come through, but any of the other cells in that grid of replicable research or robust research, where you find the same result, or maybe not, but you're at least trying to find the same result across different data sets or across different analysis packages, traditionally, journals, readers have not been interested in those findings. And I don't really know exactly what to do about this problem. I think the answer is for journals like eLife or Rio, who are trying to make it much, much easier to publish work. It's got to be good, it's got to be solid, but it doesn't have to be such a pain to publish our work. That, I think, is where we may be able to show the sort of the old guard that there is useful work that's available. It's also a great space at the moment for preprints. If you have some work that is not novel but is super solid, sling it up on bioRxiv, make sure that it's able for people to read and to cite. And I think that's one way in which we will be able to put pressure on the journals to start accepting work that is more reproducible, more robust, more useful, rather than sort of flashy and sexy and novel. I love this answer that I get from people. They don't tend to say it sort of in public forums, but at the pub and things like that, of pleading the fifth, that what if people will find mistakes in my work? I don't want to have to share my code, share my data, and basically be penalized for having made a mistake. And this to me is not really an acceptable answer. I don't think that many people get into science to say, well, all that really matters is that I get away with doing my bad research. I think most people, including everyone that's taking this course, wants to do good research. We're humans, we make mistakes, and I think it's up to us to sort of build a community that is okay with having our work checked and checking others and moving forwards when there are mistakes. Fraud, I think, is very different to the sort of honest mistakes that people make. But when we don't give people the option to show they're working honestly, we sort of bucket everything in with deliberate manipulation and fraud. And I don't think that's the case. And I think we just need to sort of be a little bit more gentle with everyone that we work with as they sort of do their research along the pathway. It does take time. It definitely takes time. Thank you all for being part of this course. This is some of the time that you need to take to make sure that your work is reproducible. There is a very selfish reason to work reproducibly, which is that once you have done your work reproducibly, changing it is very, very small. So one of my personal passions is making figures with code, because every single time you tweak your analysis, you have to remake all of your figures. It takes a little while to write some code to be able to make those figures, but it means that every single little tweak, you automatically end up with the figures at the end as you run everything, which feels super exciting. This, I think, is a great challenge. If you make your work reproducible, do you have to support additional users? I would say that you do not. It'd be great if you did. It'd be really great if it was easy for others to use your code, to try and replicate your results, to see how generalizable it is, all that kind of jazz. But I don't think at this time, where reproducible research stands at the moment, that it is on your head to support additional users. And the way in which I would recommend that you kind of make clear about your support for additional users is to have in your README file for your GitHub repository, we're going to talk about GitHub later on, just say that you are there to address any bugs that people find. If there are mistakes, you're open to hearing about them. But if they do not know how to use the code, it's up to them to find support. They may need these additional skills. Reproducible research needs additional skills, but there are actually so many tutorials and resources that are out there. This talk is going to be one of them. The online brain intensive is going to be another. And this is a challenge that we are sort of really ready to take on. My number one take-home message from this whole presentation is start small. You are just one person. You do not need to be perfect. My top tip, I am not paid by protocols.io, but I kind of should be because I love them so much. What you can see on the screen here is a screen grab of a structural neuroimaging pipeline that I have made on protocols.io. And if I just scoosh over to the next slide, I'm going to play a little video. So I have created this. It's built on top of GitHub, so you can version control it. It has a title. It has some contact information. This is how you could ask me a question. And at the moment, there are 13 steps. So what you can do is come along to it, read each of the different steps. Here I'm telling you which software you need to install. I'm also helping my collaborators in Cambridge get set up on our high performance computing cluster. You get to like mark off all of the different stages. Now, this pipeline uses a different standard for directory naming and file naming than BIDS, which you're going to learn about later on in the course. It's because I was using it before BIDS was really developed. So all I can do is write down exactly what you need to do. I can write down all the different steps, and then you can follow along with them. You can fork this pipeline, and that means that you can edit it for your own use, and you can also develop it and share them easily. Once you take a step into the exciting waters of actually having a machine go through all of those different steps for you, your coding, your analyses. And my take home here is that comments are your friend. Here's a picture of some code that I wrote. It's written in Bash, so it will run in a Linux shell. I have, and every line that starts with a hashtag, a pound sign, is a comment. You can see that I have lots and lots of comments at the beginning. The first sort of 30 lines or so are all comments. And then later on, I have a few additional comments. I have some block comments that explain what a chunk of the code is doing, and I have individual line comments that just explain what sort of specific commands are doing. Now I'm going to give you a pretty big ask here, which is to aim for 50% of your code to be made up of comments. That means that half of the text document that you create that contains all of the commands will actually also be documentation and explanations of what you're doing and why. Those comments can include links to where you found help to write the code, and they could also include people who have helped you along the way. One of those people may be the person who originally wrote the code. So it's very common in neuroimaging labs for a graduate student to arrive and for a postdoc or a senior grad student to hand them some code that they have written. And they will often say, it's not that good, it's not commented very well, I keep meaning to do more about it, but you can try and run it if you want. I think one of the nicest things that you can do to show gratitude to the person who's helped you along the way is to add in your comments and share them back. Because then next year, when a new graduate student joins the lab, they will receive a, if not perfect, at least a better commented and documented piece of code. Importantly, that imaginary next person is actually likely to be you in the future. It's you in 6 months' time when you get reviews back on a paper. It's you when you start working on a new project with a different data set and you think, oh, I have some code that does that. Where is that? Add in your comments, put your code in a place that you can find it easily, and I promise you will benefit. I just want to have like a little sassy side note here. I've received quite a lot of pushback from more senior programmers that adding comments is the wrong thing to do. That what you should be doing is writing code that's so clear to read that you don't need any comments. And I have to say, that's totally fine if you're already a good programmer. My personal experience as someone who's self-taught has been that the comments are really freaking useful. And from working with other graduate students or postdocs or master's students or research assistants who also haven't been trained officially in how to write good code and how to read others' code, add in the freaking comments, man. if you get really good, you can take them out again. But just add in the freaking comments. There are loads and loads of places online to receive help, which is amazing when you're writing your code. Stack Overflow, it took me about 5 years of trying to ask a new question on Stack Overflow before I actually succeeded in finding one that wasn't already there, that hadn't already been asked and answered. It's also a pretty cool place to find some online warriors who decide to make you feel kind of crummy for not knowing the answer. In many cases, you can ignore them. In one specific case, they have a really good point. So I want to explain this an acronym of MWE, a minimum working example. It's really hard to have someone help you if you don't give them code that they can run. So here's a question that somebody asked about the lock function in pandas. They gave some code. They were asking why it's used. And in this case, you don't need a huge amount of code in the explanation. Here's an explanation that I provided. I linked to the pandas documentation in a couple of places, but mostly I was just explaining in other words the pandas documentation. So I didn't have a massive amount of code. In this example, though, adding a line to a matplotlib scatter plot, there's some code there, but it doesn't work because there's no data frame available. There's a picture and there's some documentation. But you can see that in my answer, which starts here, I go through and I explain what all of the different measures that you'll need are. And I also provide a whole chunk of code that will create a data frame, that will create a value that's sort of got a little bit of noise in there, and that will then make this figure for you. That is a minimum working example. And when you go to ask a question, please do make sure that your question can be run as code. It makes it so much easier for the person answering the question. Next top tip is if you're installing Python, do not follow questions about installing Python from the interwebs, just use Anaconda. It makes everything so much easier. I use a Windows machine and it works really well on there. If you look online for advice on installing Python on a Windows machine, you often get advice to use a Linux machine, which is, somewhat helpful, but not really. Anaconda works across platforms. It should work for you as a Mac user, as a Linux user, as a Windows user, and it is free and easy, and that is my top tip recommendation for using Python. I want to also direct you to some beautiful galleries, which are basically these minimum working examples, showing off what some of the plotting abilities are for Matplotlib and Seaborn, which are both Python libraries. If you go along to their gallery, you can look for an image that you think, oh, that looks pretty cool. I'd quite like to be able to make an image somewhat similar to that. And if you click on each example, you will see the source code. So here's one that wants to show different error bars in a scatter plot. And as you can see on the right-hand side, if you copy and paste that whole chunk of code, especially with that big documentation at the beginning, you will be able to recreate these two figures. It's a really, really great place to start so that you can then adjust all of the figures to fit with what you would most like. Seaborn is basically building on top of Matplotlib, but trying to make the plots look a little bit prettier. They're also really lovely for lots of different sort of more complicated statistics. The Jupyter Notebook, I'm not going to spend a huge amount of time explaining the Jupyter Notebook because I know it's going to come up over and over again over the course of the online brain intensive. But basically, it's a place that allows you to write some text as Markdown, and it also allows you to put in some code. It will show you the figures in line It's an interactive way of working with the code, so it's a really great place to be able to sort of build up what you want as you're trial and erroring everything that you're working on. But it's also a great way of sharing out some text, some links, some figures, and some code for others to be able to use. The previous example was from gravitational waves. This example is from a neuroimaging paper that came out more recently, and you can go through and look at each of these different notebooks and see the different steps of the analysis. So this is a really delightful, reproducible analysis. The data is there, the code is there, you can go in and work with it. You can also make copies, you can fork these versions, and you can edit them for your own data or for your own particular needs. Here's just what one of these notebooks looks like. There's some code at the beginning. There's two different functions. You can see the output as you come along, and you, as you scroll down, it will run through all of these different steps, and it will output the figures into the browser window for you. So you can go through and you can have a look at all of these different versions. Okay, my last example of fantastic tools for helping others to interrogate and trust your analyses is NeuroVault. For a recent publication of mine with Mike Carter and Professor Sylvia Bungay, we put all of our statistical maps, so all of the results of our study, up on NeuroVault. If you click on the link corresponding publication, it will take you to the published paper. And if you go to the published paper, you will see that in all of the figures, we have at the very bottom of the caption, thresholded and unthresholded maps are available in NeuroVault at, and we have the link. If you go along to one of the links, you can see I've picked out one of the unthresholded maps for this particular test of analogical reasoning. And it allows you to actually explore the whole data set. It allows you to look to see where deactivated findings are, what the extent of activation is in a 3D volume. There's also a really nice surface viewer. But this is just a really beautiful way of sort of raising confidence in some of the results so that you're not just selecting a particular slice that looks really good for your story. You're being transparent about where this three-dimensional data lies and how everyone would be able to interact with it. It's also a really spectacular way of supporting meta-analyses going forwards. We're really near the end. My last little section is about Git and GitHub. Personally, as with coding, I found this really intimidating and it took me a while to really sort of jump on board. I want you to leave today feeling confident that you can use Git and GitHub, and knowing the difference, at least knowing the difference between the two. So this figure shows Fred, Dave, and Lisa, who are all working on their individual laptops using Git. And Git is their version control software. It's allowing them to commit changes and sort of timestamp those. It allows them to go between different changes, and it allows them to sort of revert back or go forwards and keep a track of everything that they are editing. What GitHub allows is for the three of them or more to communicate about their changes. It takes their local Git repositories, their local version control system, and puts it up in the cloud and shares them together. It allows them to merge their changes really nicely, but that's a Git feature. And GitHub allows them to very easily chat about those changes, make decisions about the changes, and sort of interactively scroll back and forward through time and through the different sections of their code It doesn't have to be code. It could also be documentation. It can be very small amounts of data, not large amounts of data, and anything that needs to be sort of version controlled. So you could write a manuscript using Git and GitHub. You can write your code. You can use it for sort of lots of different aspects. Here's a really quick overview of a traditional GitHub flow. So that blue line, that light blue line at the top, represents a master branch, and it might belong to me, it might belong to someone you've never met before, but importantly, you do not have write access to it. So you're going to create a branch of that, and you might even create a fork of the repository. I'm not going to go into the difference between those now, but you can very easily Google those and find out. When you create your branch, you have write access to it. So if you wanted to, you could delete every word and replace it with a swear word. I don't know why you'd want to do that, but you have write access. You are allowed to do that. Let's imagine, though, that you want to add in some comments or you want to fix a bug. You will commit those changes as you work with your particular branch of the repository. And when it's at a stage that you think it's useful to be merged back in, you can ask for those changes to be incorporated by opening a pull request. You cannot pull your changes into the master branch because you do not have write access there, but you can request the owner to pull those changes in. So that's why it's called a pull request. You'll then go through a stage of discussing and reviewing your code. They might ask you to add in a couple more comments. They might ask you to change a couple of the phrasings that you've got, or add in something different, or take out something, and you will make those additional commits as you're going through the review process. You will test your code and make sure that it works according to the various different sort of requirements. And once it passes those tests, you'll be merged back in. So that's a traditional sort of GitHub way of working. And I've created for you and for anyone else that's feeling unsure about how to use GitHub. This is not an exercise that's necessary for anyone that has used GitHub extensively before. This repository called Reproducible Research. And there's a whole bunch of jargon for GitHub. So repository, cloning, forking, branches, pull requests, version control, issues, release, commit, merge, markdown. And I'm not going to explain all of those here, but I'm going to encourage you to do a little bit of Googling, look through some of the resources that are already available to you, have a chat in the Online Brain Intensive Slack room, and once you feel confident that you know the definitions of some of these words, submit your first pull request. So come along to the Reproducible Research website, the GitHub repository. Check out the guidelines for contributors. Make sure, please, to follow the code of conduct, which means that we are particularly supportive of new people and people who are feeling unsure of how to begin. It is extremely important that everyone feels very welcome in this community. And you may want to join in creating a glossary. There's a page on there called glossary.md, which is our jargon busting exercise. You can check out that link and see if there's words that already have been explained. And if there are words that have not, this is your opportunity to add to those. You also might want to share resources for people that you have found useful and that you might think that others who are at the same stage as you of starting out with reproducible research and starting out with GitHub might find useful. There is a page called resources.md, and I've already added in a couple of links to help out with Git and GitHub, as well as Markdown. Check those out, but also please add some more. So finally, there are so many amazing tools to help you do reproducible research. I've put just a few of them on this slide. I know that there are so many more. Please don't feel overwhelmed. There is a whole community of people here within the Online Brain Intensive, but also more broadly, there's the Mozilla Science community, the software and data carpentry communities. There are amazing additional MOOCs on Coursera that I found really valuable. And really, I just want to finish by saying thank you, and you got this. Thanks so much for listening. I'll be available on Slack this week to answer any questions, and you can always get hold of me on GitHub at Kirstie Jane or on Twitter at Kirstie_J. Good luck. Well done.